Hello, everyone. It's a big pleasure to be with our global audience this morning and this afternoon. It's a big pleasure doing this live webinar with the collaboration of Hendrix Genetics. And we welcome Estella and Ewoud, and we're very excited about the subject today that has been captured a lot of attention in our audience. The presentation is going to be around 45 minutes. Then we're going to have a time of question and answers, and then obviously Stella and Ewood will share their contacts. And like always, this is gonna be recorded and then it's gonna be accessible in our YouTube platforms. So Stella and Ewood, welcome again, and the time is yours. Thank you very much.
Thank you. Thank you very much. Thank you for the opportunity to present here today. I hope you can all hear me. My name is Ewoud Kampschoer. I'm poultry veterinarian, and today, together with my colleague Estella Leentfaar, our nutritionist from Hendrix Genetics, we will present for you regarding Salmonella control. Today, we would like to talk about a little bit about the characteristics of Salmonella, the importance of Salmonella in the poultry industry, a little bit about clinical science and especially monitoring. Then Estella will dive deeper into the feed strategies to prevent Salmonella infections. Then I will take over again, tell you something more about sources of Salmonella and potential threats on farms. And last but not least, Salmonella control by management. First, what is Salmonella? What are we talking about? It's a gram-negative bacteria, and it's quite important to understand that Salmonella can survive with and without oxygen. So it can survive in a wide range of conditions, and that means it's also quite robust in the environment, and that will give you some challenges as well. Transmission is mainly via fecal-oral transmission, but vertical transmission is also possible in some cases. The identification of Salmonella can be done on several ways, and I will show you two examples of how it could be done. One of them is based on the O and H antigen, and if we base them on the O antigen, we always talk about group A, B, C, and D, and on the screen today you can see just a few examples. And for the poultry, the most relevant ones are, of course, Salmonella typhimurium and Enteritidis in this case, but there are a lot of different serotypes for Salmonella. So this is just a brief idea. But the groups that sometimes are mentioned in papers or publications or whenever you read about Salmonella, if they talk about group A, B, C, and D, it's only based on the O antigen. That's important to remember. If we're going to classify the different kinds of Salmonellas a little bit better and then based on characteristics of the Salmonella, we can also talk about host adaptive and non-host adaptive serotypes. So we're not talking about the groups anymore, not based on the O antigen, but we're talking about more or less the characteristics of the different types of Salmonella. We have host adaptive Salmonella types, and they cause systemic infection. They can create high mortality, and a few examples of those are Salmonella gallinarum or Salmonella pylori. These are relatively rare at the moment, or at least in Europe, due to eradication programs. If we talk about non-host adaptive Salmonellas, we are usually talking about gastrointestinal infections. And that means that those are infections in animals, but also humans. So they are not very specific for one host. And they can be in a bird or usually we talk about birds. It can be subclinical and then colonization inside the intestine. So that's important to remember. So then we have Salmonella Enteritidis and typhimurium. So completely different stuff that you can see in your house depending on the serotype. So it's important to understand what we talk about if we talk about Salmonella. The host adaptives, those have huge negative impact on animal health, but the non-host adaptive can also have a huge negative impact on human health. And that brings us directly to Another important reason why we are having this topic for today, because why does salmonella matter? First of all, salmonella for the public health, food safety. Salmonella is like the second most common foodborne infectious in the EU after Campylobacter. And that means a lot of people get sick from that. I will show you later in the presentation, next slide, how that looks like in Europe. But salmonella can also have a huge economic impact on the egg and poultry production and on the entire industry. The third reason why salmonella matters is for regulatory pressure. There are more and more rules in each country. It doesn't matter where you are, but the regulation regarding salmonella is everywhere getting more and more and more. So and that has some good reasons. So I'm not complaining, but it's important to realize, and that's why we need to pay more attention to controlling salmonella. And last but not least, salmonella can have an impact on your reputation, and it can have a risk for your market access if you want to export eggs or day-old chicks or poultry products in any kind. If we look at the map for Europe, you can see the amount of cases of confirmed salmonella cases in people. So this is the main reason why salmonella is important in poultry, because of the effect on human health. And this is a huge underestimation. So these are only confirmed cases, but you can imagine that not everybody who has a health challenge will go directly to the doctor and get a confirmed diagnosis. So the numbers are probably much higher than we realize in this research. But you can see that everywhere in Europe and everywhere in the world, salmonella is causing a challenge for human health. What can we see in our chickens? As I explained already a little bit, it can vary a lot. It really matters what kind of salmonella you have in your flock, what type of bird you have. Are we talking about broilers, layers, day-old chicks, young birds, older birds? It can be from extreme mortality all the way up to no clinical symptoms at all. And especially that last one is important, because if your birds look healthy, it does not mean that they are not infected with salmonella. And that's why monitoring is very important. It matters to monitor, because you can do it in two different ways. I'm starting. You can diagnose during clinical cases. So if you have high mortality, but that's only if you really have a problem for your health of your birds. But if you want to control the human health, it's very important to have routine monitoring inside every chain of the poultry industry. So in the hatchery, you can sample or monitor chick paper, and of course in the house with boot covers. Do this regularly and do it in a good, proper way, because if you don't monitor in the correct way, it's just a waste of money and time, and it doesn't help you. If you have a proper monitoring system, it can also help you improve your health status of the flock and your access to the market and all the points that I mentioned above. Before we're going to dive deeper into the ways how you can control salmonella biomanagement, I would like to give the floor to Estella to tell you something about salmonella control in feed.
Thank you very much. So indeed, there is always a major risk via feed as well. And one of the examples is, for example, contaminated raw materials. And this really much depends on what kind of raw materials are we talking about. I will come back on that later. But also, for example, dust accumulation is a very important one. Because this accumulation is also a source where bacteria like salmonella can grow on and then recontaminate the rest again. That can happen both in the feed mill as well as in the feeders. Next slide, please. Yeah, thank you. So when are we talking about risky raw materials? Well, for example, there has been already a lot of research done and shown that animal-based products are having a higher risk, almost 4 times higher risk compared to plant-based, although it also depends which type of plant-based we are talking about. And further, grains have a lower risk compared to oil products. And oil products, then we talk about our protein sources mainly. So soya, sunflower, rapeseed, all the byproducts from the oil industry have a higher risk for Salmonella contamination compared to the regular grains like wheat and corn. They have tested the amount of Salmonella in compound feed, so in final feed, in both 2017 as well as 2023. And you see it's relatively low. It's close to 0.3, 0.4%. However, this is every delivery towards your farm. So if you calculate it over the total lifespan of a bird, it can still be quite accessible. It's also noted, but I couldn't find the number directly, but it was stated that Salmonella enteritis, where we just were talking about causing also human health concerns, is rarely found in feed. But nevertheless, other Salmonella sources can be. It's also shown that, for example, 2 Salmonella per gram of feed is already sufficient for infection. So you don't need to have so much Salmonella to cause infection in our laying hens. Then depending on storage, because a lot of raw materials are stored for a very long time, but you will see that dry storage is mainly important, and a long dry storage is decreasing Salmonella a little bit, but it will not prevent Salmonella entering via the feed. Next, yeah. One other, a more recent case, I would say this has happened 2 years ago in the Netherlands. There was a very big research because there was a high outbreak of Salmonella. And at the end, it was caused by eggshells using as a raw material. So eggshells, from a circular point of view, can be used as a calcium source in our poultry diet. But if heat treatment is not properly done on eggshells, it will have also a consequence, because it can cause Salmonella to Salmonella between the poultry. Therefore, processing of eggshells is very, very important, and it also requires proper monitoring to prevent having infections in your other flocks. Strategies for preventing. First of all, starting with the raw material management. I'll also show you in the next slide a little bit how this is being done. Zoning, so dirty versus clean, is a very important one. So for example, if you do any treatment, which I will explain later, it's very important to separate. And then it's not only separation in the feed mill, but also separation on, for example, the trucks. So you cannot mix trucks transporting treated feed versus trucks not transporting treated feed, because then you can still already have a recontamination during your transportation. Dust control, I already mentioned, dust control is a very important one, not only for dust explosions on the feed mill, but also from a Salmonella point of view. And then, of course, there are, but Ewoud will explain further in detail, but controls of pests, rodents, reptiles, very important in some situations, but also pet management, like your cats or your dogs, they can also contribute to recontamination at the feed mill. And further, the moisture control is very important, because Salmonella needs to have a moisture environment to continue growing. Next slide. Thank you. So here you see just possibilities where contamination can happen. In general, what research has found is that there's a 1.5 times higher occurrence in before heat treatment compared to after heat treatment. So after heat treatment, you will see much lower values of Salmonella. What you can see here is, of course, the delivery. I already mentioned that oil products, so soya meals, have a higher occurrence compared to grains. But between soya and grains, you can have a recontamination during storage, or, for example, also during weighing or during mixing. During feed manufacturing, well, you can have an acid treatment. I will explain you later. You can have a heat treatment as well. And then afterwards, we have storage of the feed. During storage of the feed, but also especially during cooling down of the diet, you can have a final recontamination. Why do I explain cooling down? Because if you do a heat treatment on your diet, you need to cool down your feed afterwards, and then it's very important to use filtered air. If you don't use filtered air for cooling down your product afterwards, you can already get a recontamination directly because the feed is still very hot and humid. And this is also shown a little bit on this slide, where you see, of course, during intake, there can be already some salmonella. During storage and weighing, there can be salmonella again. During the mixing, the cooling, as I mentioned, very important one, because normally after conditioning, you need to cool. And then we are talking about this post-heat process, which have lower values of salmonella, but can already start recontamination. So it's very important to have this pre-heat process as the dirty zone and to have the post-heat process as the clean zone, and to keep it separated totally. When we talk about treatment, heat treatment, you can have several thermal treatments for reducing salmonella. Most used ones are either pelletizing, then we are talking about a high temperature for a short duration, or we are talking about sanitizing, which I will explain more in detail. And sanitizing, then we are talking about around 80 degrees for at least 60 seconds. You can see on the right side, you see how the process is going. So here you first have mixing. After mixing, you have conditioning. Conditioning means increasing the temperature of your mesh diet to, well, recommended 85 degrees, but in general, 80 should be sufficient. And afterwards, you have a time retention barrel where you will just maintain the duration. In this case, on the right side, they're using 150 seconds. It should be at least 60 seconds to remove salmonella completely. And afterwards, cooling down. As you can see on the right side, they're using HEPA filters, which are comparable with hospital filters, and therefore preventing bacteria recontamination afterwards. Then you have storage, and you should totally have separated storage and transportation of your heat-treated feed. It's also important, a lot of people are taking samples afterwards to confirm that the heat treatment is done properly. What you, however, see is salmonella is kind of shocked because of this thermal treatment, and therefore it's not very much recoverable, which means you can show that you have a negative sample, but if you will wait a few days, you can get recontamination or reoccurrence of the salmonella. And therefore you can have a false negative sampling if you do it directly after the treatment. There's also an acid treatment. I think most well known is most likely a formaldehyde treatment. Works very effectively, not allowed in all countries. So when formaldehyde is not allowed, we do see that other organic acids in general are not strong enough to kill down salmonella. They are strong enough to prevent any recontamination after a heat treatment. So in those cases, often this combination is being used. And then you can talk about an organic acid mixture of different acids. What is the goal of such an acid? The goal is actually to penetrate the cell wall of the salmonella and therefore reducing the salmonella. As mentioned, mainly to prevent recontamination, so to prevent survival and especially multiplying of the salmonella. If salmonella occurs after heat treatment, which can also happen, then either the timing is wrong, so you did not hit the 60 seconds, it was most likely a little bit shorter, because what you often will see, it will take some time before the first part of your batch is on the right temperature, and then you need to make sure that also this batch is on the right temperature and the right duration. Recontamination after the heat treatment is a very important one, as we have spoken, but also warm and humid storage, for example. In some climate, it's very difficult to cool down properly enough. And that's, of course, having a risk for recontamination as well. But also improper wet cleaning. What you often see is people taking care of their feed mill, they do a wet cleaning when they talk about heat treatments, just to make sure at least once a week that they don't have any recontamination possibilities. But if you do a wet cleaning, you also need to make sure it's totally dry before you start running your machines again. If it's not fully dry, you can already get some recontamination, as Salmonella likes to grow on wet surfaces. Well, this is just some advisors from the GMP Plus International and from the VFAC. And there you can see for your own reference, okay, how much samples should I take to make sure that I have a proper monitoring? And you will see for pure line and grandparents birds, this is much more compared to general brooders or layers. And if a Salmonella type is positive, then it's also required to do serotyping, as already a little bit explained by Ewoud. And then some specific feed safety limits. Basically, it should be 0% or close to 0%. As you can see for laying hens, so commercial diets, then they say, okay, the ones causing human health should approach the 0%, but all the other salmonellas can be up to 1% present in the diet. If you look to broiler breeders or layer breeder diets and even turkey diets, then it's not allowed to have any salmonella inside. Then I give the floor back to Ewoud.
Thank you, Estella. We talked about a little bit about what is salmonella and of course how to control it by the feed. But what we can do, if you can, I would say differently. You can control your feed and you need to do that. But there are a lot of things you can also do on farm to control your salmonella status. So before we talk about that, I think we need to discuss the biosecurity zones, because it all starts with a proper biosecurity plan. And I would challenge you all to think about your own farm or operation and see if you have clearly designed these kind of zones. In this example, we have 4 zones, but it can be in practice even more and sometimes less, but more is better. And I will explain you later why. The green zone is the zone where the birds are. That's the zone that you want to keep clean of all the diseases, and in this case, salmonella. The red zone is the outside uncontrolled road. So that's a public road. You cannot have any control about what's going on, who is driving there, and that's out of your control. But then we have 2 different zones in between, the green and the red one. First one is the buffer zone on the dirty side, in this case, the orange one. And we have a buffer zone on the clean side. And what it means, every time when you cross a line going to a different zone, you should take a certain biosecurity measurement. And by applying these measurements all together, you try to keep your birds free. And it's very important that these changes of zones are very clear defined. I will first show you some biosecurity measurements, but you can imagine between the red and the orange zone, you can, for example, create a rule or a fence or a reception or anything to get restricted access. So not everybody is allowed to go onto the dirty buffer zone, the orange one. By just controlling who is entering your farm, with a very easy fence or some signs or anything, you can already improve the biosecurity. If you go from orange to green, the light green, you can, for example, implement a shower. Everybody needs to shower before you can go to the buffer clean zone, so the light green one. By showering, you minimize the risk of bringing in bacteria or viruses on your hands, in your hair, in your nose, or whatever. And it will make sure that people will change fully their clothes if you have proper clothing on farm. And then if you go from the light green to the green, you go to the birds, and then you have the last barrier where you need to do a boot switch, and you need to wash, dry, and disinfect your hands. If you follow all these protocols, and if the zones are properly defined and easy to manage, you can reduce the risk of getting an infection of Salmonella or any other type of disease on your farm. On top of that, Estella already explained to you how you can get lower risk in your feed. But even on farm, it's quite important that your feed truck and egg truck are in a separate road from the people that are clean. So you need on farm, you need a clean and a dirty road for the transport of dirty trucks from the outside road. I will show you later an example. And of course, I mentioned it already here, and I will mention it later as well, rodent control. Because you can define your zones, but as long as all the rodents can go from red to orange to green to light green to green, you still have no control about Salmonella. So monitor all your rodents, make sure that the environment around the houses, in the houses, around your silos is clean, and that it's not a nice environment for rodents to be. If we talk about biosecurity measurements, a lot of people ask me, what is the best measurement we can take? And there's no silver bullet in this story. It's important that you think about combining biosecurity actions, because to be honest, it doesn't really matter which of your measurements stopped the bacteria or virus from coming in, as long as one of them did. And maybe today measurement 1 will work, and tomorrow measurement 2 will do a better job. As long as one of the measurements is doing a proper job at that time, the chances that your virus will come into the farm is less likely. So I try to represent it with this picture. You don't want to get fire on the right side, you want to stop the fire. And it doesn't really matter where it stops, as long as it doesn't reach the other side. So every day it can be a different measurement. Combining measurements will create a much stronger biosecurity management on your farm. This is a picture of an example of one of our pedigree farms where we defined 5 different zones. And you can see that the houses are in green, the houses where the birds are. And by defining these zones and showing this to all our employees on the farm, it's easier to train them, it's easier to explain them why it's important, and it's easier to stick to the rules as well. Because if you only make rules for paper, but it's very difficult to follow them in real life, it's of course not getting the purpose. On this farm, you can also see that we have a dirty and a clean road. You can see that the light green, for example, is the clean road where the employees can enter the farm. So after the shower, they can go on the clean road, they go to the greenhouses, and they switch again their boots, they switch their clothes, they wash their hands, and they are allowed to enter the farm. But a feed truck or any other delivery or removal of feces or any kind is being done from the other side of the house. So you can see that there's a road as well on the outside of the farm. So the dirty and the clean road will never cross. If you want to define your zones on farm, this is a very nice strategy. It's by defining the zones with colors. Because if you create a very easy biosecurity friendly environment, it's for everybody, although they want to do it good, as long as they don't know what to do, it's very important, very difficult to do everything every time the right way. By giving each zone a different color, it's very clear if you're allowed to walk in a certain zone with a certain color. This way, you can see on the right side a perfectly clean boot switch. Very simple, very cheap, very easy to use, and you won't make a simple mistake. I think it's very clear for everybody that green is for the left side and yellow is for the right side. If you talk about environmental reservoir for Salmonella, it can be in a lot of different things. As mentioned by Estella already, dust, dust, and dust. Dust is very difficult to control, but dust can definitely contain Salmonella, especially if you have an infection in the past on your farm. The litter can be a reservoir for Salmonella, feces, but also cracks in your house and concrete, your rodent population on your farm, and of course your feed silos and feed mills and everything related to feed can contain still some Salmonella. All these different topics should be addressed if you have a Salmonella case in the past. If you don't pay attention to all these details, you will have a big risk of reintroducing a Salmonella. This also means that the outside area of your farm is potentially a risk for Salmonella if the rodents go into your house. And it's not only the rodents, but it's also been shown in different researches that a lot of houses still have trouble with leaking rainwater coming in or a spillage of water coming in from the outside, and they can bring in bacteria or viruses under the doors. So pay attention to all these details to improve your biosecurity. Nevertheless, people are still a big risk. So train your people well, tell them what they need to focus on, and together with that, you can create the best biosecurity zone on your farm to reduce the Salmonella risk. On top of the Salmonella control by management, there's of course also vaccinations available. Depending on your strategy and depending on your risk and biosecurity level, you need to decide what kind of vaccination program could be right for your type of bird or your type of risk. To give you an example, if your biosecurity is high enough, it's, for example, not needed to vaccinate for Salmonella because, you can control the risk by combining a lot of different biosecurity measurements. If your biosecurity is less, it could be helpful to give your bird some extra immunity with vaccination. There are a lot of different types of vaccinations. In general, it can be live or killed, and in practice, most of the times, a combination of live and killed is being used. But just a reminder, vaccination is on top of biosecurity and not instead of biosecurity. It cannot replace a biosecurity measurement. It can give you extra protection for the birds, but please invest in your biosecurity first. The take-home message for today, to summarize, Salmonella can survive for a long period in the environment. That's why it can cause a big risk on farm or everything that is brought into the farm. And I forgot to mention, but also egg trays or other materials that go onto a farm can be a big risk for Salmonella because the Salmonella can survive on those surfaces. Salmonella control is important for both human and poultry health. Feed can be treated to reduce the risk. Estella explained us 2 different ways to do that. And a good biosecurity is a combination of different biosecurity measurements to reduce the risk of an introduction of a disease. And with that, I would like to thank you all for joining this webinar today. And if there are any questions, please do not hesitate to contact Estella or me.
Ewoud and Estella, thank you very much for the presentation. And we will share this video maybe the next week in our social media. And thank you very much. And let's see you in the next one. Thank you, guys.
Thank you.
Thank you very much. Have a nice day.
Thank you. Thank you, everybody. Bye.